Okay, thank you everyone for coming to my talk. I'm Henry Arenas-Castro. I'm a first-year postdoc at Yale University at Jen Cochran's lab. And this work was done together with Kesh Cochran. So yeah, let's talk a little bit about like monkey flowers that probably you are already quite familiar with them by this time. But first, I want to acknowledge that this research has been conducted on indigenous land, the unsettled land of the Mohera, Machatuket, Pequot, Eastern Pequot, Eschaktikok, Golden Hill, Pagokset, Niatik, and Kinyapik, and others Algolokian-speaking peoples. I pay my respect to their ancestors and their descendants, who continue cultural and spiritual connection to land. I also acknowledge the legacy of slavery and enslaved African people whose labor was exploited for generations to help establish the business of Yale University. So the main topic of my current postdoc is about drought adaptation in monkey flowers, and I really hope to share some exciting results with you next year. So meanwhile, I have been just like playing with some fun data, and that is actually what I'm going to present to you. So this is really preliminary, very preliminary data, and more than just telling you a story that is complete, I would really appreciate your insights as well about like what other things I can do just to figure it out, like this kind of like paradox of these species pairs. And first, I just want to say that all the praise of this research should go to Kesh, that was a brilliant undergraduate student that decided that biomed is closer to his heart, and all the blame you can just dump it on me eventually. So I just want to bring to your consideration like this classical continuum or panmixia and complete isolation that we have seen in different talks. Traditionally, we have considered species as those that have completed the process of speciation, that they are effectively reproductive isolated, and we can see cases like some sunflowers that even though they might have like some gene flow, they have strong reproductive barriers. But quite often we see other situations, and I think that given the talks that I have participated in this conference, applies for most of the cases when we have like different populations or what some taxonomies consider a species that they have high levels of gene flow. So thanks, Grace, for the pictures, by the way, as is the case of the Texas frogs, and that has been also one of the regular talks of this conference. But on the other end, there are a lot of like panemitic populations that individuals kind of freely interbreed between themselves, yet you can differentiate like two different groups of morphological species, if we want to call it that way. And that is kind of a paradox, like how, despite the homogenizing effects of gene flow, two different populations of species can keep their distinctive like traits or characteristics. So one potential explanation for this, or what has been invoked in most of these cases, is invoking the role of natural selections that can express in different ways, directly, for instance, as reinforcing selection, but also in the context of two populations of species that are adapted to contrasting environments. Local adaptation can incidentally contribute to the coexistence of these interbreeding teams, species, populations, as we want to call it. So in this diagram that deliberately I took it from a paper that is bilingual in Mandarin and English, just to highlight that wonderful science is done around all the world in different languages, and just to cherish that. We might have like two populations and that are like different chromosome regions that are freely being exchanged between the two populations of the species, but some regions of them are more resistant to gene flow because they harbor genes that underlie adaptive phenotypes, or phenotypes that are also important for processes like sexual selection. So in those areas of differentiation, you can see that the effective gene flow is little, and the neighboring areas, just because linkage disequilibriums, they are dragged in the different statistics that we can use just to characterize like population or species differentiation, as one of those being FST that we use. And I'm so happy of having attended like Sheila's talk about like these seed eaters, semilleros in Argentina, because it's pretty much like this case in which they are quite homogeneous along the genome because they hybridize quite often, but still there are like some peaks of differentiation. So in a genomic view of speciation, this is a pattern that is becoming more common and common as we delve deeper into these like paradoxical cases of incipient species. So yeah, for my talk, I was recently introduced over the last year to the Mimulus or Mimulus guttatus complex, the monkey flowers, yellow monkey flowers, that is an amazing group that has been studied from an ecological and genetic perspective, and there are still a lot of phenomena to uncover in this charismatic group. And this study is framed mostly in northern California, where two of these species co-occur, Mimulus guttatus and Mimulus glausescens. They are quite different, either in the field or in the glass house. You can say that the glausescens species grows upright, they don't branch out that much. The stem is waxy, quite different to the one of guttatus, but also the leaves at the nodes, they are fused, opposing leaves are fused. They look like a single leaf that just cover the two axes. And previous studies have uncovered or characterized different reproductive barriers, and none of them are contributing to total reproductive isolation. So you can see here the total reproductive isolation that is not necessarily close to one to be complete. So there is like room for a lot of gene flow. And also when we study patterns of differentiation or genetic distances, we can say that this is the genetic diversity within the species of glausescens, and this is the genetic distance within the species of guttatus that has two different clades, northern and southern. But when we check what is the genetic distance between guttatus and glausescens, depending what is the clade that you are focusing on, in northern and southern, is lower than the genetic the differentiation or the distance that you can find within the same species. So yeah, that challenged a little bit the idea of like a good or complete species, even though that they kept all their characteristics in the field. Oops. So it brings us to the question of like, what maintains this phenotypic differentiation in Mimulus glausescens despite these high levels of gene flow? Oh, again, oh my God, I fixed it. But imagine that there are some dots here, and I was working with glausescens that they are restricted to this area in Northern California, and they are mostly in these two counties of California. And it co-occurs with Mimulus guttatus in blue, is in the area of sympatry, but the species itself, guttatus, distributes or occurs along all the west coast of the United States. And there is a third species that also co-occurs with them, but it's a highly selfing sulfur species. So in the first approach that we took, we analyzed like the genetic structure of this group of species. And when we just consider only like two ancestry components, it clearly differentiates Nasutus, that is the sulfur, versus glausescens and guttatus. And also these traits of guttatus, sorry, the Nasutus are consistent with other studies that have detected asymmetric gene flow from the sulfur Nasutus to the outcrosser guttatus. And we also found one individual that phenotypically looks to us as a hybrid individual, and indeed looks very hybrid here. When we consider three ancestry components, we can see that there are a component that are mostly restricted to glaucicens, so supporting that there might be like some genetic structure or some genetic differentiation of glaucicens. And when we consider like four ancestry components, we see one of those that are mostly restricted to guttatus and glaucicens. And it gets a little bit more messy considering 5 of them. But despite this, when we do a principal component analysis, it clearly differentiates nasutus as expected in the first principal component, but also the two clades that are the southern and northern clade of guttatus. And on the second principal component, we clearly can tell apart the closer to which the samples of glaucicens belongs compared to the sympatric guttatus. And the patterns of genetic diversity are quite similar within and between the two species, no major surprises. But the differentiation is pretty much like minimum compared to outgroup of the group. They are highly differentiated with the group, but between them, they are quite homogeneous. But still, there are like some peaks of differentiation measured as the population branded statistics that is based on FST. So trying to narrow down where might be like those genetic regions contributing to this phenotypic differentiation, I tried to analyze the top 1% of the PBS hits that it corresponds to almost 2,000 of them. Then I narrowed down considering only the bottom 1% against these values that might bear the signature of positive selection. And I narrowed down to 89 hits across all the chromosomes. And finally, I focus only on the hits with low genetic diversity, probably because they have gone through a strong selection. And I identify 10 hits in 3 different chromosomes. So the hits in these chromosomes point out to transcription factor, CCR4 This one is an interesting one because it corresponds to a non-chromosomal inversion that differentiates annuals and perennial individuals in Mimulus guttatus. So it's a segregating inversion that probably is not related to the phenotype that we are interested. But this big surprise of here, that in a total different chromosome, we found that a difference of unit of the same transcription factor complex are contributing or have the signature of selection that can contribute to this phenotype, that is really unlikely to happen. And I think that there is a lot of potential of this particular master regulator to be involved in this. The thing is that this is highly pleiotropic. It's involved pretty much in transcription in general, and it's lethal, their deletion. But the interaction with the different subunits has been used by different plants just to acquire new functions, like, for instance, response to far red light and nodulation. And probably there is a lot of opportunity to explore what is going on with this transcription factor. But probably it's going to be a little bit messy given how pleiotropic might be. So just the insights that we are gaining from this preliminary data is that, yeah, there is a lot of like gene flow between these two species, yet there are like some genetic structure. Potentially, the genetic factor has a polygenic landscape, this adaptation, because we have hits across all the genome, but there might be one or few genes of large effect that can be also like responsible for the differentiation that we are seeing in these species pair. Thank you, everyone. Gracias for coming. And yeah, and might not have time for questions, but happy to talk in the coffee break or later. Thanks.
Hello, I'm Anoob Prakash. I'm a PhD student from University of Vermont, working in Kerala lab. And today I would like to talk about genetic architecture of local adaptation in a species with a history of introgression. So what is local adaptation? It is the population adaptation selection that varies geographically, and local genotypes tend to have higher performance or fitness at their native sites compared to an introduced site. And it is important because local adaptation is responsible for shaping species diversity, ecological speciation, and also to facilitate range expansion. And traditionally, local adaptation is studied using a reciprocal transplant experiment, wherein you take a genotype from the native environment and plant them at their native site and an introduced site, and vice versa, and try to see if there's a fitness trade-off across the two environments. And the genetic basis of local adaptation, or genetic architecture, is traditionally explained by two alternative models of how alleles confer fitness in contrasting environments, antagonistic pleiotropy and conditional neutrality. Let's dive deeper into what those terms mean. When alleles confer some advantages in one environment, mostly the native environment, and disadvantages in a different environment, that is antagonistic pleiotropy. And the example here looks at mean fitness on the left side and looking at different sites. It shows that the fitness is higher at the native site and lower at the introduced site. Another example is taking the locus of a flowering probability for Bucera stricta. We see again Montana does better in Montana and worse in Colorado, and vice versa. So that is the trade-off we are talking about. Conditional neutrality, on the other hand, again have advantages in one of the environments, but not really a trade-off in the other, so sort of stays neutral. And here it's looking at another two different locuses for the same fitness trait. And you can see that locus 1, the allele A, is advantageous at site A, but neutral at site B. But locus 2, vice versa, there's nothing happening at site A, but at site B is at an advantageous potential. So that makes some sense, right? So that's the whole premise of what I'm trying to talk about genetic architecture of local adaptation, and want to see how it affects adaptation and how standing genetic variation can help us understand if there are any trade-offs in the future as we have warming climates, so antagonistic pleiotropy, or if they're staying neutral and may come under selection in the future, so conditional neutrality. And it is important because once we know about this, we have more control and understanding about how to predict the species adaptation to warming climate. Talking about warming climate, this is my study system. Red spruce doesn't really like hot climates. Coniferous tree species adapted to cool montane environment found throughout the Appalachian. And the map on the left shows that the green distribution. Towards the north, it has a continuous distribution. Towards the south, not so much. Too much warm temperature, fragmented towards the high elevation spots called as sky islands. But it also has a history of range expansion since last glacial maximum, wherein since the glaciers melted, the range has expanded northwards. And it is also predicted that in the future, the adaptation will be much drastically reduced, and that habitat will be not suitable at the southern part of the range and have lower genetic diversity. So we have established red spruce have issues. So I wanted to understand what are the issues and how we can tackle them. So one way to do that is look at the genetic data. The spruce genome is huge, so we didn't sequence the whole genome. Instead, we looked for whole genome exome capture of 340 individuals across range. And while doing this, we found out that along this range, they split into 3 genetic clusters. The one in the continuous distribution north, denoted by the yellow triangles, is the core. The fragmented distribution in the warmer south, denoted by the blue diamonds, is the edge, and the one between these two range edges is what we call the margin. You don't have to keep track of all those names, but that's the 3 clusters we have. In addition to that, we also grew them in 3 common gardens at Vermont, Maryland, and North Carolina for 2 years. And we collected a lot of phenotypic data. And we also looked at the climate space these mother trees occupied, and they were occupied more cooler climate, as I explained. Common gardens, when we looked at the most northern part and the Vermont and the southern part, North Carolina, Vermont is bit cooler compared to the North Carolina, still way outside what the trees were used to experiencing. So you'll be looking at these two gardens, where you will use the North Carolina garden sort of as a space for time substitution of what warming weather might look like. And out of all the traits we measured, we will only be looking into survivorship and biomass. We have a lot of other traits measured, but we are going to use only these two traits to have relative fitness trait. And based on that, we are going to run two GWASs. And here, basically, Manhattan plots, we generate GWAS for the relative fitness at home and away, and the SNP, the height of the SNP is basically the significance of how well that SNP is associated with the trait. So we are looking at the significance of the SNP as well as the effect sizes. So effect size is depending on how many genetic variant of an allele you have, how much it contributes to a trait variation. And so that's what we look at. On the right side, we see the relative fitness. The effect sizes can either be positive or negative. The sign doesn't matter other than to help us categorize. It doesn't mean a positive or negative effect size means a positive or negative effect on the trait itself. It's more of the impact that variant has on the trait. Now that we have that information of the SNPs and effect sizes, we can, and we are using something called beta coefficient or the effect size, we can categorize them based on these GWAS results into the GA categories I explained before. So for antagonistic pleiotropy, if the effect sizes, if they are significant and have differing sign at the differing gardens, so if it's a positive at home and negative at away, or negative at home or positive at away, that means antagonistic pleiotropy, the trade-off. If they are significant at one side and neutral at the other side, then it's conditionally neutral. So for home, if it's significant only at home, then conditionally neutral at home. And if it's only significant at away garden, then it's conditionally neutral at away. In addition to these two categories, we also have universally beneficial wherein it's positive in both garden sites. Right, so we have our structure. Let's go into some very, very preliminary results. Some of you might have seen this before. Don't be alarmed that it is not yet updated. But yes, we look into relative fitness, and as expected, we have a higher relative fitness at the northern Vermont garden and the southern North Carolina garden. For the sake of keeping track, we will just call the Vermont garden as home and the North Carolina garden as away, because it's a warmer site. And we see the same differentiation in the different genetic clusters as well. And you will see there's a bit more of an erratic high relative fitness in margin. There's a little bit of a story there. Hopefully I can touch upon it later. Okay, so I ran the GWAS for relative fitness for at both home and away, got the significance, got the effect sizes, so we have everything we need to establish our genetic architecture categories. Once we have the SNPs and the effect sizes, we can plot the home results of the beta coefficient from the home against the away side, and the line that runs along the 0 point, that is where the neutral SNPs are going to lie, and on either side of this 0 line is going to be the significant or selected SNPs. So if that's the case, if you look for conditionally home, conditional neutrality at home, then we should expect to see them beside these two, along beside the 0 line on the x-axis for the home, and we expect to see conditionally neutral away beside the 0 line on the y-axis for the away side. So we got the results. We have a lot of SNPs that are under conditional neutrality at home and away, and we also get a lot of universally beneficial SNPs. And you will see a little bit of antagonistic pleiotropy right along here. To get a clearer picture, we can separate them into its own different categories, and we get a clearer picture of all the universally beneficial SNPs, the antagonistic pleiotropy, and the two conditional neutrality. And you will see they are only significant at their home site for home and away site for away. So now we have the categories for the local adaptation. What next? So looking, I talked about in the title introgression, and I never brought it up again, right? So let's think about that. So we have black spruce, a sister species that has a much wider range and distribution in the North America, and it has a overlapping range with red spruce. And during in this overlapping region, there's a lot of hybridization and introgression happening. Right, and so the local adaptation in red spruce is not only just contributed by the standing genetic variation, but also by this adaptive introgression with black spruce. So we want to understand if black spruce introgression, how it affects the GA categories, right? So we got some black spruce samples in the allopatric samples outside the range of red spruce, and we did genetic PC, and we saw that there's a separation with the margin having more connection towards the black spruce. And we looked at genome-wide scans for selection between these two species, red and black, and tried to use these SNPs on the SNP bits along the PC1 of the selection scan to have the locus differentiating along this selection axis. And we plot the categories on them, and it shows that fitness loci does show a significant shift based on the genetic ancestry, but I still don't know what is driving that. It could be black spruce, it could be red spruce, but that is the question mark where I am left it at right now, but hopefully some updates soon. So to summarize, most of the genetic architecture of local adaptation in red spruce was found to be conditionally neutral, so suggesting that genomes that are under selection in the past might not be the same regions that might be under selection in the future. And a significant number of universally beneficial SNPs were found, which is positive news because that means these SNPs will be beneficial irrespective of growing conditions, something I would not have believed before I'd done this for red spruce. And a lot of, not a lot of antagonistic pleiotropy, but that could just be because it's first 2 years early fitness trait, so it might not be able to completely capture all the antagonistic pleiotropy or trade-offs that we might see in a long-lived tree species. And going forward, I would also like to understand what the introgression is playing in the GA categories, because in a different study, we have found that introgression does truly play a major part in adaptation to, assisting in adaptation to different selections. So that is very interesting part I am looking forward to. And I would like to thank all my collaborators, all my lab mates who have helped me go through all this analysis. I am not smart, I just had a lot of help. And thank you, and open for questions.
Good afternoon, everyone. I'm Ethan Newman, and I am from Rhodes University, situated in the eastern part of South Africa. And so today I'm going to be talking about a model used for studying pollinator-mediated speciation within the angiosperms. And so this model is based on extracts taken from two quite important publications. And so one is, well, for both of these publications, you're probably very familiar. So one is Pollination Within the Phlox. And so this is by Verne and Karen Grant in 1965, and another by Stebbins in 1970. And so during the mid-2000s, extracts from these two publications were taken and was coined as a Grant-Stebbins pollinator shift model of floral evolution. And so one of the sort of key quotes from this model is, as you know, Stebbins' most effective pollinator principle, which states that since selection is a quantitative process, the characteristics of a flower be molded by those pollinators that visit it most frequently and effectively in the region where it's evolving. But what's really interesting is that even many years later, very few studies looking at pollinator-driven speciation actually looks at how efficient pollinators are. And I just want to go into a little bit of detail with regards to this model. And so, say for example, we have, as you can see, we have South Africa over there, and we have the hypothetical range of this particular plant species over here. For example, if you can imagine that on the east part of this range, this plant overlaps with the distribution range of this particular butterfly, and this indicated by the stars over here. But then also, on the other hand, you could argue that honeybees occurs across the entire range of the plant. And so where the ranges of the honeybees as well as the butterflies overlap in this region over here, as you know, honeybees often, well, at least in South Africa, they are not very good pollinators often because they carry a lot of self-pollen and often not in large quantities. If you had to compare that with butterflies that carry a lot more outcross pollen of higher potential quality and as well as quantity. And the expectation here is that you would have evolution towards the east of the range or adaptation towards butterflies. The result there would be you'd have larger flowers adapted to the morphology of that butterfly, whereas to the west of the range, those flowers are adapted to honeybees. The flowers are a lot smaller. And so ultimately, you would expect to find that when they make secondary contact through floral isolation, you have gene flow barrier occurring and potentially a speciation event. And so that kind of summarizes this Grant-Stebbins pollinator shift model. And so the study species that I've chosen basically to try and test this model on is this plant called Brunsviga gregaria. And this is a plant that I've been working on for the last 2 years. And so a lot of the information I'm giving you is a little bit descriptive, but still very interesting. And what makes this plant interesting is that it lacks a corolla tube, as you can see in the image. And so nectar is held with what we call a nectar chamber. And so that means that pollinators of varying mouth parts are able to access that reward despite there being an enormous amount of floral variation within a very small range in which this plant occurs. And so you could actually argue that this is a pollination generalist. So what we did is across 9 localities in an area about 90 square kilometers, we sampled floral traits. So we looked at tepal length, pedicel length, style length, and flower number. And so these are all functional traits associated with pollination. And so you can imagine something like tepal length and pedicel length would be associated with pollinator attraction, and style length with how the pollinator fits the flower. And so we've just imported this into a very simple cluster analysis that provides us with 3 putative ecotypes. More interesting is that the styles, they vary significantly. And all that I've done here is that you can see on the y-axis I've got the functional length, that's functional style length, and then the different populations. And you can see that there's an increase in the mean and the standard errors surrounding that. And so if you had to cluster those according to those groupings, they're all statistically different from each other. And so you have your short style, intermediate, and long style ecotypes. So with that variation in mind, 3 research questions come to mind. And so you can ask, do distinct pollinator assemblages associate with geographic variation in floral traits, these generalist pollination systems? And within those assemblages, are particular pollinators more efficient than others? And then just lastly, asking whether there's ongoing phenotypic selection on floral traits within each of those ecotype groupings. So for those first 2 questions, I conducted pollinator observations, or rather my students conducted these observations. And what they basically measured was looking at both visitation rate as well as the frequency of contact with the anthers and stigmas. And so if you have to take the product of that, this gives you pollination efficiency. And so what we basically found in these populations, were especially at the long style populations, are these long proboscis flies. And they can often have very long proboscis up to 7 centimeters in length, and the flowers' stigmas are often equally as long. But there was also others like pollen collecting bees, bee flies, honeybees, as well as butterflies, and they match the reproductive parts of the flowers to varying degrees. And so what we've basically done is I've constructed this bipartite network, and this is based on visitation rates alone. And so on the left, you can see these are the different localities, and I've just colored them based on the different ecotype groupings. And on the right, you can see these are all the different pollinators. And so if you look at only visitation rates on its own, you can see that this network is quite nested, so many connections between the nodes, with the thicker lines, obviously, the thicker the lines, denoting higher visitation rates. And so when we apply a clustering function to this basically what we find is not a very strong association between our different modules that has been formed by this clustering function and the different ecotype groupings as per the cluster analysis. However, the story changes quite a lot, and also consider that many studies only look at visitation rates, which tells you that if you don't incorporate contact or incorporate pollinators that make contact with reproductive parts, you might get an inaccurate picture. And so this is based on pollination efficiency. And so you can see that the network looks very different. In fact, many of the connections that were present within the previous network are actually lost. And the only reason for that is because although those pollinators visit the flowers, they rarely make contact with the reproductive parts. And so again, if we apply a clustering function to basically have a look at, the degree of how this network can be broken up into modules, there's a much closer association. And so the darkness of these individual triangles, squares, they basically indicate how efficient a pollinator particular is and its contribution towards that module. And you can see that medium-sized bees, which include honeybees and some solitary bees, they contribute mostly to this particular module over here, and that's associated with the short style flowers described in the beginning. Intermediate modules, those are associated with swallowtails and skipper butterflies, and then long proboscis flies are very important in the formation of this particular module over here, which is associated with long style plants. And you can see a very strong association there with the most effective pollinators associating with those different ecotypes. And so you can see that I've basically brought up this particular slide again. And so what I've done here is that I've overlaid this slide with the styling with the mean functional body lengths of the pollinators present within each of those populations. And so you also see that in this figure, there are different shadings or rather intensity of blues. And so the more intense your blue is, the more effective that pollinator is within that population. And you can see that there is a rough association between the stylings of the flowers within the population and the pollinators that are most efficient. And so you can see at the intermediate sites, butterflies associate the best with the styling there. Same with long proboscis flies at the long style site. So they have the darkest colors and best associate in terms of stylings with those flowers. And that's obviously, if you had to look at the relationship between the two, it turned out to be significant. And so lastly, we were also interested to see whether there's ongoing phenotypic selection on those floral traits. And what we did is, for each of those different ecotype localities, we conducted a phenotypic selection experiment, so using the Landin-Arnold approach. And we looked at our functional floral traits, so we looked at styling, tepal length, as well as flower number. And what we basically found at the short style site is, firstly, we found correlational selection between style length and flower number. We also found significant directional selection on flower number, as well as you can see, stabilizing selection on style length. Not illustrated here, though. For our intermediate style site, again, we found significant selection or positive directional selection on flower number. So this is something that is often documented within these studies. So the more flowers you have, the more seed set you have, and so stronger directional selection. And then we've also found then stabilizing selection on tepal length. We didn't find any selection at the long style site, which was really interesting. However, when we compared selection between all of these sites, most of these comparisons were significant, indicative of divergent selection on floral traits between these different ecotypes. So just to summarize the study, so as you can see that there is still much to be learned, especially with regards to how individual functional pollinators within each of those sites are responsible for driving selection. Just to highlight again, we had 3 floral ecotypes, And those basically differed significantly in flower morphology and associated assemblages. And we basically found that within those assemblages, although they are diverse, the most efficient pollinators, they seem to be driving floral adaptations, as evidence from the trait matching that you've seen from that relationship between functional pollinator length and style length. And it's also that the most important pollinators are likely to drive ongoing phenotypic selection on floral traits. Future research may include conducting a reciprocal translocation, and within that reciprocal translocation, doing single visits and measuring seed set from those single visits from different functional pollinators within those different ecotype populations to look at the contribution of each of those to fitness and local adaptation. Yes, and so this is just to thank the many people that have helped in the field and farms that have allowed us to work on their property and some of the different funding bodies. Thank you for listening. And I'll take any questions that you guys have, of course.
Did you check if there was a difference in anther stigma distance between the different ecotypes?
So you're asking in terms of ecogamy?
Yeah.
That is something that we did not check, but those anther stigma distances, they are very, very similar, and the reason for that is because they're protandrous. There's first a male phase and then a female phase, and so the position in which those anthers are basically release the pollen, the stigmas basically receive is exactly the same. Yeah. Yeah. Any other questions?
Can I ask just like a botanical question? Like, do these ones mask like other Brunswigia species?
Sorry, can you just repeat that?
Yeah, do they mask? Like, do they all bloom at the same time?
Oh, yes. Yeah, so all of them do bloom at the same time. What's really interesting is so the area in which they occur are relatively close to each other. So it's sometimes even within 2 or 3 kilometers of different ecotypes even. And so how it works in that part of South Africa is that if there's some heavy rainfall that comes through, all of them bloom all at the same time, which gives us an opportunity to potentially do translocation experiments and that sort of thing.
I might have missed this, but is there any difference in flowering time between any of the ecotypes?
Oh, it kind of answers the previous question. So they all flower at the same time depending on the
But
rain.
there's no like difference in like the schedule within a bee?
Oh, flowering, do you mean flowering duration within an ecotype?
Yeah, like, you know, I mean, even differences in a couple of days can really reduce the amount of overlap in flowering length by.
No, it's more or less the same time, eh? But, yeah, I'm happy to answer any other questions. Thank you.